Hi, everyone, and welcome to today's webinar, Enhance LNP Development, Cryo-TEM, and Automated Image Analysis Tools, presented by Dr. Mandy Janssen. We're excited to have you with us. We'll be reviewing the advantages of Cryo-TEM for imaging lipid nanoparticles in their near-native state, explore the wide variety of critical quality attributes Cryo-TEM can reveal, all in a single dataset, and discuss the applications of Cryo-TEM from R&D all the way to CMC and regulatory support. Before we begin, let's go over a few quick housekeeping notes. The webinar will run for about 30 minutes, followed by a 15-minute Q&A session. You can submit your questions in the Q&A box in your toolbar, and Mandy will answer them. If we don't get to your question during the session, we'll follow up with you next week. A recording of the webinar will also be sent out next week. Now, it is my pleasure to introduce our speaker, Scientific Director of Characterization, Dr. Mandy Janssen. Prior to joining NIS in 2017, Mandy spent almost 15 years in the electron microscopy field. She obtained her master's degree in biomedical engineering from the Eindhoven University of Engineering in the Netherlands, and her PhD in molecular pathology from the University of California, San Diego, where she worked with both light microscopy and transmission electron microscopy. Please join me in welcoming Mandy. Mandy, over to you.
Hello, thank you for joining. Today, I will show you how Cryo-TEM imaging and advanced image analysis techniques can be used as an exceptionally powerful tool to quantitatively analyze LNP formulations, not only during early development, but throughout the entire CMC pipeline. Let me start with introducing NIS, short for Nano Imaging Services, before I step into what Cryo-TEM is and how we use it for LNP analysis. NIS was founded back in 2017. Over the years, we have specialized in two service areas, namely high-resolution single particle analysis to aid our clients in their structure-based drug design, and nanoparticle characterization, where we use Cryo-TEM and advanced image analysis tools to characterize quality attributes of gene and drug delivery vehicles, such as size, morphology, integrity, aggregation, payload distribution, and presence of impurities. We have a total of 6 microscopes divided over 2 coasts. 4 are here in San Diego, and 2 are in Boston. Our San Diego site is also GMP compliant, and this is the site where we analyze many different types of formulations for either late clinical or commercial phase or aid in sample analysis for regulatory submissions. Why transmission electron microscopy? In short, because of the very high resolution images we can attain. So in transmission electron microscopy, electrons are extracted from an electron source under high vacuum and at acceleration voltages between 80 and 300 kilovolts in general. After electrons pass through the specimen, the scattered electrons are focused by a series of electromagnetic lenses onto a camera or a direct electron detector. Because electrons have a much shorter wavelength than light, the resolving power of a TEM is much greater than that of a light microscope, which allows us to distinguish much smaller details. For example, TEM allows us to visualize individual viruses and LNPs, and we can distinguish the double leaflet structure of a bilayer. And in many instances, we can even resolve protein entities or complexes on the surface of these viruses or LNPs, if present. We can also look at discrete proteins and nucleotide strands within a sample throughout the background. So here are some examples of nanoparticles that we frequently look at NIS. We typically look at particles up to about 1 micron in size, and these can be proteinaceous in nature. They can be virus-like or virus particles, liposomes, extracellular vesicles, LNPs, but also inorganic particles such as gold and iron nanoparticles. We can use these images to evaluate the quality attributes such as size, aggregation, integrity, morphology, lamellarity, and payload distribution. We also can look at complex formation, we can do epitope mapping, as well as determine high-resolution structure for particles that are homogeneous in size and morphology. One thing you may notice here is that the particles in the panels on the right are dark on a bright background, whereas the particles in the panels on the left are white on a dark background. This really has to do with the sample preparation technique. On the left, we use negative staining TEM, and on the right, we look at particles in vitreous ice, or Cryo-EM. Whether we use one or the other technique really depends on the sample type, as well as the question we would like to answer. So in negative staining, 3 microliters of sample is added to a small 3 millimeter metallic TM grid covered by a continuous thin layer of carbon that is made hydrophilic by charging its surface. After leaving the sample on the grid for about a minute, the sample is blotted off and a drop of heavy metal stain is added. This is then also blotted off, and then the grid is air dried. Negative staining provides a lot of contrast and is especially useful to, for example, visualize really small particles or look at, for example, integrity of larger nanoparticles. However, staining and drying artifacts cause lipid-containing particles to lose their original shape and size. And because you are staining the outer surface of the particles, there is no information we can extract from the interior of these particles. And you can see in this image how messy these particles look. So Cryo-EM is a much better option to preserve liposomal particles. And in this technique, we also use 3 microliter of sample, which is added to a similar 3 millimeter metallic TM grid. But now there's no continuous carbon layer, but the carbon layer on top of the grid contains evenly spaced holes. And since the sample is not stained and the contrast is only derived from the sample itself in Cryo-EM, the sample particles are best imaged over these holes to limit addition of any background noise due to the substrate. You can see here in this image that it is much more difficult to see small proteins in ice compared to in stain. These are the same sample. But the LNPs in ice maintain their natural hydrated shape and size, and you can even see the interior of these particles, showing compartments that are more dense than the surrounding buffer, which is the lipid phase, as well as compartments that are as dense as the surrounding buffer, and that's the aqueous phase. Looking at this LNP sample in both Cryo-EM and negative stain TEM, it's very clear that TEM or Cryo-TEM is really the superior technique for this type of liposomal sample. So as I mentioned before, in Cryo-EM, we apply 3 microliters of sample to a grid, then blot most of it away until a very thin layer is left, and then rapidly plunge freeze this into liquid ethane that is cooled down to liquid nitrogen temperature. This plunge freezing step is done using a robot, and we can control the temperature, the humidity, the blotting time, and the blotting force, and this makes the grid preparation process reproducible. After the grid is then transferred into the microscope, the grid is usually screened at a very low magnification to check for areas with optimal ice thickness and ice quality. And increasingly higher magnification images are then acquired to get a complete picture of the sample and to set up targets for automated data collection at high magnification. So the high magnification images that you see here are then used to assess particle size, homogeneity, aggregation, payload distribution, morphology, thickness of the bilayer and or PEG layer, particle integrity, and distribution of surface protein if present. And this can be done all within a single data set. Of course, there are some challenges when imaging LNPs. The particle concentration we generally require is a minimum of 10 to the 12 particles per mil, which for typical LNP formulations translates to about 10 to 30 micrograms per mil lipids. I'm not sure about how much this is for mRNA concentration. The presence of sugars, such as sucrose or trehalose, can be detrimental to the particle contrast, as you can see in the top right images. So this is a 10% sucrose image showing LNPs with rather low contrast with respect to the background. After a buffer exchange to remove this sucrose, the particle contrast is significantly improved. For optimal image contrast, the sugar concentration is best kept below 5%. In many instances, LNP formulations do contain 10 to 15% sucrose, And for those cases, we typically image the LNPs in high sucrose first, evaluate the contrast, and then if needed, we reduce the sucrose content simply by diluting in a buffer that doesn't contain sucrose immediately prior to grid preparation. And this usually is very helpful. Another obstacle are certain LNPs that like to stick to the carbon substrate instead of going into the hole. And to get around that issue, we can use either other grid types like lacy carbon, where the low amount of carbon drives particles into the holes, or we can play with the charge we add to TEM grids to make them less negatively charged or even positively charged. After optimizing the grid preparation step, we acquire images that provide us with enough particles to perform a sizing and morphology analysis if that's requested. So here is an example of how the particle segmentation looks for an image after we run the automated particle tracing tool right there. So the segmentation you can see is the green traces. These particle traces are then subsequently fed into an algorithm that then calculates sizing metrics, such as area equivalent diameter, minimum and maximum thread diameters, circularity, and their respective d-values, aspect ratio, span, etc. So you can also see that this is an example of such a size distribution, and you can see that if we make 3 different preparations of the same sample, the size distribution looks very reproducible. We can perform then also a particle morphology classification analysis based on any predefined features. So in the case of mRNA LNPs, we can, for example, look at fully dense particles like this one, particles that appear to consist of 2 or more compartments, which is called blebbing. In this case, these are the blebs. And we also can determine if these aqueous blebs contain any grainy material, which is presumably the mRNA. We can also look at modeled appearance of the dense phase in LNPs, which may be indicative of the presence of mRNA intercalated with the lipids, as this modeled appearance that you can see here is usually absent in particles that are not loaded, which you can see here. We have now developed an automated particle classification algorithm making use of computer vision techniques that enable us to recognize particles with certain features, so with and without blebs, as well as blebs that are empty versus blebs that are loaded with nucleotide. And we can currently do that with at least 90% accuracy. The models are still being improved, but in the end, this will be very useful to understand the morphology within certain size populations of particles and how these subpopulations may possibly change during development or manufacturing. I will now give an example of why and where in the pipeline quantitative analysis of LNPs can be extremely useful. Obviously, LNP size and morphology depend on lipid and buffer composition, mixing and downstream processing methods, as well as possibly the targeting moieties that are being used. Currently, standardized methods to evaluate particle physicochemical characteristics are DLS to measure the size. Oftentimes, zeta potential is assessed as well. And then the total mRNA encapsulation is measured by a ribogreen assay. This does leave morphology. And so far, published studies show ambiguity in whether unilamellar particles are more potent than bleb particles or the other way around. But in general, the field seems to agree that morphology is an important quality attribute that does need to be characterized. And this is where Cryo-EM comes into play, as will be shown in the following example. Okay, so during a collaboration between NAS and Phosphorex prepared several LNP formulations, and they basically all consisted of a standard 4-lipid component formulation. But they were prepared using different mixing platforms. They used the T-Mixer, the Ignite Plus, the Jet Mixer, and the Sunshine. In addition, the formulation that was prepared by the Ignite system was evaluated after dialysis into low salt versus high salt, using dialysis versus tangential flow filtration, and using a 6,000 nucleotide payload versus only 858 nucleotide payload. And lastly, we also checked one of these formulations after 10 weeks in storage at 4 degrees Celsius. First, let's have a look at the results when various mixing platforms are used. So here are 4 representative images of LNPs generated by each of these mixing systems. And by eye, it may not really immediately be clear that there are differences, but when performing a quantitative size distribution analysis, you can see that although the Ignite, Jet Mixer, and Sunshine are all very close in size, the particles generated using the T-mixer are slightly smaller. Interestingly, a much larger difference is observed when we look at particle morphology. While, again, the Ignite, Jet Mixer, and Sunshine show very similar proportion of particles with bleb formation, the T-mixer, on the other hand, shows much fewer of these particles, namely 27% as opposed to the 40 to 64%. And this is a huge difference that would never have been observed if you only run DLS. Next, we look at a standard preparation dialyzed into low salt buffer and loaded with mRNA, and that is 6,000 nucleotides in length. We compare this standard preparation to a similar formulation, but dialyzed into a high salt buffer instead of a low salt buffer. It's here in the top right image. And we compared the standard preparation to the same formulation, but after using TFF instead of dialysis, which is this image. And then we looked at a standard preparation, but changed to a shorter payload length, namely 858 nucleotides instead of the 6,000. And this is what you can see in this image. You can see the size distributions of all 4 preparations, and you can see how these small changes do affect the size, except for when changing payload lengths. And in that case, it stays around 63 nanometers. It probably depends on the mRNA you're using and the exact lengths, but in this case, it stayed the same. This is something you can possibly see doing DLS. However, what you cannot see with DLS, again, is the change in morphology. And it's incredible to see how a high salt buffer eliminates the number of bleb particles, going from 46 in low salt to only 1% in high salt buffer. The other 2 conditions also clearly affected, but a little less. And knowing that this morphology or bleb formation is closely linked to LNP potency, I think it's easy to understand how important direct visualization of an LNP formulation actually is. Lastly, I'd like to point out the use of Cryo-TEM when looking at stability at different storage temperatures or perhaps in time. So here you can see a fresh sample on the left that is less than a week old, and the same LNP sample after 10 weeks of storage at 4C in the absence of any sucrose. You can clearly see that part of the population has not changed, but there are particles present with enormous blebs, multiple compartments, and most importantly, big aqueous compartments. So this is yet another example of where visualization of LNPs can be extremely helpful. So quantitative analysis using Cryo-TEM images is reproducible, and this is shown here for 2 of the formulation conditions I presented to you in earlier slides. Analysis in triplicate for these showed similar respective size ranges as well as similar fractions of particles with bleb formations. So in the first formulation, size was consistently around 43 nanometers with about 1.5% particles that showed bleb formation. The second formulation showed the size of the particles that was consistently around 60 nanometers with a fraction of bleb particles that was about 10%. And then for those of you interested, in this experiment, Phosphorex also measured the average diameter of particles for each of the conditions I presented to you in the last few slides using DLS. And here you can see how well the size measurements correlate between DLS in blue and Cryo-EM in orange. Not surprisingly, the size measured by DLS is generally larger due to measurement of the hydrodynamic radius and its general emphasis on larger particles. How much this difference is likely depends on the particle type you're imaging. Now, if you like to see your particles in 3D, for example, to assess whether you have antibodies bound to the outer surface of your LNPs, you can do either immunogold labeling, or you can do something that's called electron tomography, where we collect images while tilting the sample within the microscope, for example, from -50 to +50 degrees, and then acquire an image every 2 degrees, and then back project all these images into a 3D volume. This can give you a little bit more information than a 2D projection image can. For example, in this liposomal doxorubicin product, if you're not familiar with the sample in Cryo-EM, you may recognize the doxorubicin crystals within the liposomes, but you may wonder what these round densities here actually are. And then there's some faint elongated structure in between here, and it's not directly clear from the 2D projection image what this is. So when you do electron tomography and you look at the 3D reconstruction, you can start to appreciate that this dark round density is actually a top-down view of your doxorubicin crystal within the liposome. And that faint elongated particle is likely a flat disc-shaped lipid particle that's intercalated between the other liposomes. So to summarize, Cryo-EM allows us to look at particles in their near-native hydrated state using direct visualization at the per particle level. So only as little as 3 microliter of sample per grid is required to acquire a full dataset, and one full dataset can provide information on multiple quality attributes at the same time. In addition, Cryo-TEM analysis is not affected by impurities, unlike, for example, DLS, where even dust particles can throw off the signal. And lastly, I hope that I have shown you that using automated image analysis tools can make Cryo-EM analysis quantitative, unbiased, and reproducible. And as a final takeaway from this webinar, I'd like to briefly point out the use of Cryo-TEM throughout the entire CMC pipeline, not only for LNPs, but for many other types of nanoparticles as well. For example, Cryo-TEM is an exceptional good tool to characterize 2-component systems, such as viral particles in the presence of adjuvant, or to aid early development of novel vaccines that you can see here. During later phases, Cryo-TEM can be used to assess, for example, payload distribution here for AAV, size or morphology, when it comes to evaluating the drug or vaccine during scale-up of the production volume, during batch-to-batch comparison, or when evaluating the stability of a drug product. And with this, I'd like to thank you for your attention, and I'm happy to answer any questions.
Thank you, Mandy, for that great presentation. Before we move to our live Q&A session, I wanted to direct your attention to some content in the chat. First, we have our email address, which is nischr@nanoimagingservices.com. Please reach out if you would like to discuss your particular LNP project, or if there's additional questions after the webinar you'd like to chat with us about. Link to our LNP white papers are also in the chat if you'd like to learn more about our capabilities. And so now we are going to go to the live Q&A portion. If you have a question, just as a reminder, you can submit them to the Q&A box in the toolbar. And our first question, Mandy, is about the number of particles that we include. So how many particles are typically counted during data analysis for blebs and sizes? And then a second question with that, and at what magnifications?
All right. Yeah, thank you. Thank you. Very good questions. So the magnification that we use for sizing and bleb morphology counting is usually 73,000x. This is about 0.2 nanometers per pixel. This gives us a good number of particles per view, but it also gives us enough contrast or enough details of the particles to actually see if particles are blebbing, yes or no. The number of particles really depends on what kind of study we're doing. If it's a typical R&D study, we usually count or size a minimum of 250 particles. If you're looking to validate these kind of methods, we count a lot more particles, usually I would say a minimum of 1,500, but it really depends on the homogeneity of your sample. The more homogeneous it is, the fewer particles statistically you need. The more heterogeneous, the more particles you need. But that's something we would figure out for that particular study. The particles of the examples that are shared in this presentation, for this experiment, we used between 1,000 and 1,500 particles for each experiment.
Great. Our next question is about talking more about blebs. So is there a consensus yet about whether blebs are beneficial or detrimental to particle potency?
Yeah, that's another good question, and I wish I had the answer to that. From what I hear or read in the field, there's not much of a consensus yet, but more and more it appears that morphology is important. And there's a couple of papers that are already out that came out that says bleb formation can enhance potency. And I think a paper from Peter Collis actually showed that if the mRNA was in the particle is present in the aqueous phase, so in the bleb, that makes it even more potent. So I don't know, it probably is very dependent on formulation.
Still a debate. The next question is about visualizing things on the outside of these LNPs. So would it be possible to visualize protein ligands, such as antibodies, using cryo-TEM?
Another good question. Yes and no. So first, it depends on the size. Entities like a Fab would be way too small for us to see with the naked eye. So if we talk about antibodies, technically it would be on the small side, but if you have enough antibodies conjugated to the surface, we may be able to see it. So we have done projects in the past where LNPs were conjugated with antibodies, and you can definitely see patches of protein on the outside of the LNPs. And especially when you do then something like electron tomography, in which we make a 3D volume of the sample, you can really start to see the kind of the coding around these particles. The bigger the entity is, the easier it is to see, and the higher the conjugation efficiency is, the easier it is for us to see. Yeah.
Speaking about things on the outside of these particles, so is it possible to see the PEG corona on the surface of the LNPs?
All right. So the PEG corona, we cannot directly visualize. It is something we can directly infer it from the images. So if you have a lot of liposomes with pegylated lipid on the surface, they tend to not touch each other. So if you have a high enough concentration of LNPs in the image and you can see them at regular distances from each other, then you can kind of infer that half that distance between two particles is consistent with the PEG layer. Yeah. And especially if you then have like antibodies bound to your pegylated lipid, you can start seeing a gap between your protein and the outer surface of the LNP.
Right. So it's visualizing, but more of a indirect, right? So we can infer. So that's more of a qualitative look at these particles.
Exactly.
All right. Next question. Are there any differences in sample concentration requirements for negative stain TEM versus cryo-TEM imaging?
Yes, to stain, you need a much lower concentration of particles for. So when we're talking about LNPs still, any particle, I guess, it's about 100-fold, 10 to 100-fold difference. So if you want to bring your sample into cryo-EM, you need 10 to 100-fold more lipid or protein to properly visualize that. Because obviously a lot more gets blotted off in cryo-EM, and it doesn't stick to the carbon layer, it has to go into holes, so you want a higher concentration of particles.
Right. And that's definitely why when we meet with clients, right, and talk about projects, that we go over which workflow and all of those things are taken care of during that. Do we have any data on the relationship of these characteristics and the transfection efficiency and the toxicity of these LNPs?
Unfortunately not. So we do a lot of work with clients, but it's all confidential, and a lot of times we don't get all the information on these particles. So we wouldn't be able, I would love to. If somebody wants to collaborate on that, we would love to do that, and we'd love to learn more.
Right. The particle size that you presented appears to be significantly smaller than what's typically observed for IJM or Ignite platforms. Could you share the core size of the T-mixer? No, the bore size, I apologize, used in your process?
Unfortunately, I cannot.
Okay.
So Phosphorix made those samples and they can likely share it. They're probably happy to share it. I can tell you that for all the 4 mixing platforms, the same flow rate was used to get the same kind of dimensions out of each of these particles formulations. I'm sure Phosphorix can shed more light on that.
Awesome. Could we use cryo-TEM to define payload like mRNA integrity?
I think in a certain way you can. So there's a couple of papers that we base this on. One is the Brader paper from 2020. And so particles that do not contain any mRNA typically have a lipid phase that's evenly dense or an aqueous phase that's evenly not dense.
Mhm.
Whereas particles that are loaded with nucleotide typically so they can have an aqueous phase where the mRNA has been migrated into this bleb. And in that case, you see a very grainy background in the bleb. So that grainy material inside this aqueous compartment is indicative of the presence of mRNA. When we talk about the lipid phase, it's a little bit more difficult, but we often can also see a mottled appearance in the lipid phase, which could be indicative of the presence of mRNA. But unfortunately, since there's no standards that we can use to really test this, we also don't know what the minimum amount is of mRNA that would be present for us to actually see it.
Right. Continuing on with payloads of LNPs, so is it possible to distinguish between mRNA loaded and unloaded LNPs with cryo-TEM?
That's about the same question, right?
Mhm. Or can we, like, so basically the just distinguishing, so we can do that qualitatively, right? And we can also do it quantitatively. Okay. Next question. What kind of data analysis or software was used in analyzing size distribution and bleb features from our cryo-TEM images?
It's a good question. So both require a different type of tool. For both of them, obviously, we pre-process the images to equalize the contrast, flatten the images, normalize the contrast. But then for the sizing, we use computer visualization where we make use of edge detection. And I cannot share all these details. We don't use ImageJ or any commercial software. We make our own software in-house. We have our own software development team. But once the particles are traced, then we can automatically derive all the values using MATLAB from these particle traces. When it comes to morphology, we make use of AI models and that we're still continuously improve to be able to use for basically all LNP formulations.
Okay. I know you shared some of this data, but maybe we can just summarize it for this question. So how well does our cryo-TEM data correlate with DLS or other orthogonal methods?
Yeah, so the slide that I showed on the size showed that the Cryo-EM data correlates really well with the DLS data, with the size measured by both techniques. So but the only difference is basically that the particles, the size measured by DLS is a little bigger in this case than measured by Cryo-EM because of the hydrodynamic diameter and because if you have bigger particles present in your formulation, they scatter more and they enhance the signal. But this gap, I can imagine that that's kind of dependent on the formulation or the particle type you're looking at. But in general, like the correlation is really good.
Good.
And we have looked at the correlation between, or we are looking at the correlation between the payload distribution between cryo-EM and other techniques, but we're still working on that. So, and at some point, if there are standards and there's like a gold standard method for this, that would be awesome.
Right? We all would be grateful. All right, next question we have, are the LNPs that stick to the grid potentially, are they, the ones that stick to the grid, are they potentially different than the ones that are basically over the holes or in the ice over the holes?
I doubt it. So we usually look at both. So we look at particles on the carbon and in the hole to make sure that there's not something weird going on. We have done some R&D work where we look at particles like the size and the morphology on top of the carbon versus the holes. And for LNPs, we have not found big differences there.
Okay. Yeah, the follow-up question to that is there is a potential for bias. So yeah, we asked that. I think we have time, or it looks like there's only one more question. If anyone else has any questions, feel free to throw them in the chat. Do we expect to see any differences in LNP bleb formation as a result of any freeze-thaw cycles for LNPs?
I don't know. It depends on the formulation, I think, but definitely we're always asked to be very careful with these preps. We're always asked not to vortex. We're always asked not to freeze thaw them. So I can imagine that these are very precious samples. And so usually we make sure we don't do that. And Sarah, I think you can also comment on this. Like we often get fresh samples from clients and we make sure we image them the same day. Yeah, I can imagine it changes the morphology.
And I think one of the slides that you showed about the stability studies, right? So we can look at the time and the storage and buffer conditions. All right, we had one last one, and then we'll wrap up the webinar. Have we ever looked at imaging transfection of cells with LNPs to study the mechanism of action?
No, we have not, but that's a great question. I would love to, but for that, I think you need sectioning, and
Right.
that's a technique that we currently not have in-house.
Right.
That would be awesome to look at.
All right, and with that, we'll conclude our webinar on LNPs. I'd like to give a huge thank you to Mandy again for a great presentation and for answering all of your questions. Thank you to all of you who attended today's webinar and contributing. If you weren't able to get your question in today, or if you're watching this later, please feel free to email us at nischr@nanoimagingservices.com. That'll be in the chat again. And just as a reminder, a copy of this recording will also be sent to those who attended and registered. So with that, we'll say goodbye. Thank you again for attending, and we look forward to chatting with you all again soon.